Well, what a pleasure to be here and join with you in a whole series of considerations of innovation, of discovery, and in various ways of precision medicine. The point I'll make is really a simple one. Scientific wellness is going to transform healthcare. And I'll spend my entire lecture trying to convince you that's true. When I went to Caltech in 1970, I was really impressed with the enormous complexity of both biology and disease. And indeed, it reminded me of this fable of the elephant and six blind men, where each of the blind men felt a different part of the elephant and argued it was a spear, it was a fan, it was a trunk. And of course, it was all of those and much more. What was interesting at the time is we didn't have a conceptual framework for dealing with complexity, and later that came to be systems biology. But what was also clear at the time is that if you consider the elephant a complex system, you really had to invent a whole series of tools that could let us more thoroughly define that elephant as a biological systems. And I had the good fortune to be involved in a series of 5 paradigm changes over the last 45 years or so of my life that inevitably dealt with complexity, but they also brought us to our current insights with regard to systems medicine and something I call P4 medicine, which I'll define a little later. The first of these you've heard about, bringing engineering to biology, developing actually 6 different instruments that allow us essentially to read and write DNA. What this did was opened up high throughput biology, and certainly it was the entering path for big data and its analytics, and you will see how this is obviously a central part of medicine today. The second paradigm change came about as a consequence of developing the automated DNA sequencer and getting invited in the spring of 1985 to Santa Cruz, where Bob Sinsheimer, chancellor, had assembled 12 people to consider whether or not the Human Genome Project was a good idea. And we came to 2 conclusions. One, it was possible, though at that time, certainly technically difficult. And 2, I was astounded to see that there were a split in the group, 6-6, those that opposed and those that were in favor of the Human Genome Project. And indeed, going out into the community in 1985 and 6 and 7, it was striking to see that probably, at least initially, 80% of the biologists opposed the Human Genome Project. And of course, the institution that really opposed it was the National Institutes of Health. And in fact, it didn't change its mind until it became obvious the Human Genome Project was going to occur. But there was another aspect of the automated DNA sequencer that was really interesting, and that is to execute on developing that instrument, we had to bring together engineering, chemistry, computer science, and good molecular biology. And that made me realize that cross-disciplinary biology was absolutely going to be critical in the future, because if you had leading-edge problems, you wanted to be able to point toward the development of relevant technologies and subsequently the analytic tools you needed to analyze the data. And I tried to persuade Caltech in the late 80s that Caltech ought to have such a department, and the chemists and the engineers and the physicists all were in favor of that, but the biologists utterly opposed it. And I'll leave it to you to figure out why that might be. But Bill Gates made it possible to move to the University of Washington and start there. The first department, we called it molecular biotechnology, and it was really transformational. It developed the first 2 key techniques for the then newly emerging field of proteomics. Phil Green developed the software that fueled the entire Human Genome Project. Geir van den Eng developed a fascinating multi-parameter high-speed cell sorter, and we actually developed there the inkjet synthesis technology that Agilent later commercialized. But it was clear at that time that the next step I wanted to take, which was to develop an Institute for Systems Biology, was really running into the bureaucracy of a state university. And it was for that reason, in 2000, I resigned from the university and started then the Institute of Systems Biology. And it was the first to begin considering systems science, systems approaches both to health and disease. And of course, the systems approach to disease very quickly got us into systems medicine and P4 medicine, predictive, preventive, personalized, and participatory. And I'll just say, in putting together these 5 paradigm changes, I think the major lesson I learned is innovative new ideas can almost never emerge from old institutions. Rather, you have to create new entities that give birth to these new kinds of ideas. And that is going to turn out to be true for all 5 of the paradigm changes we've talked about here. So what exactly, how do I see systems medicine? Well, I'd say there are 2 fundamental aspects to it. One is the idea that you have to deal with complexity by generating an enormous amount of relevant information. So the idea that in the future, each individual is going to have dense and dynamic data clouds, virtual data clouds of billions of data points, is going to give us the ability, after integration and modeling, to optimize wellness and actually avoid disease. And we'll show you that this hypothesis is indeed a reality today. We've already done experiments that show how successful this approach can be. A second point about systems medicine, of course, is the idea from these kind of data, you can actually extract the biological networks that fuel, in an information sense, development, physiology, aging, and when they become disease perturbed, generate disease. So if we can actually distinguish a disease perturbed network from its normal counterpart, you can begin to get, as we'll see in a moment, fundamental insights into the disease mechanisms, and you'll begin to get new insights into new approaches to strategies and to therapies. What is, of course, interesting about biological networks is they operate at really all levels of information, at the chromosomal, at the molecular, at the organismic levels, and they seamlessly are integrated to transmit this biological information. So at the end then of 2006 or so, we clearly had a picture of what systems medicine was, and we're beginning to get an emerging picture of what P4 medicine was. And of course, the idea that I started thinking about then was, how are we going to take systems medicine and P4 medicine and bring it into the healthcare system? And the challenge really was very similar to the elephant analogy, namely, we needed to develop a whole series of technologies and strategies for systems medicine that didn't exist. And that was when, in 2007, I had the really good fortune to meet the Minister of Luxembourg and Minister of Finance at Luxembourg, and he had decided to convert Luxembourg from a 90% dependence on financial services to bring in biotech and healthcare, and asked us to write a proposal. And we did, and we proposed to do for them many things, which we were largely successful in doing. But what they did for us was really transformational. They gave us $100 million over a period of 5 years to invent the strategies and technologies for systems medicine that could never have been supported by NIH, because many of them were too daring. There was not sufficient kind of evidence, and in fact, there wouldn't have been the money to really carry them through effectively. So Luxembourg let us do that, and it really placed medicine then, by 2013, at this really interesting tipping point where we could do very powerfully many things that were in time going to really transform medicine. What I proposed in 2012, 2013 was one way to bring P4 medicine to the healthcare system was to think about a longitudinal study examining 100,000 normal individuals with regard to wellness, and doing it via the creation of these dense and dynamic data clouds, and we'll talk more about that in just a moment. And of course, as you all know, as we've heard many times already this morning, Obama, in his last State of the Union address, pushed the idea of precision medicine, never really defining it. And I'll argue that a lot of what we're going to talk about today is really the very essence of precision medicine, and it's really ripe for application to pediatrics, and we'll talk about that at a different point in time. So the technologies we developed had to do with automated DNA sequencing at a different level, had to do with creating reagents that I think in 10 years will replace monoclonal antibodies. I'll say more about those in just a few moments. It pushed the idea of single-cell analysis and how it's going to reveal a lot of the secrets of development, physiology, and of disease. And of course, we developed at ISB a series of tools that, together with other people, that enabled us to do targeted proteomics on the one hand and much more global proteomics on the other hand. And at the same time, we developed these strategies for carrying out systems medicine, and I'll talk about those that are colored in red. So we'll talk about how important it is to understand disease from its very inception all the way through to the end, and it's virtually impossible to do this in humans. We started doing it in models, but we have ideas about how to do it in humans now. We're interested in making blood a window into health and disease, very powerful new techniques of let us think about how to get effective protein biomarkers. And then finally, we'll talk about these dense dynamic data clouds and the longitudinal studies that both let us transform wellness and will in the future let us transform our understanding of disease and everything. So let's talk about the dynamics of disease. We started studying many years ago prion-induced neurodegeneration. So you know when t equals 0 in the onset of the disease, because you inject activated prions into the brain of the animals, and then you can compare over a 22-week period the transcriptomes of diseased animals and their normal counterparts to identify the transcripts that vary from one another. And of course, those can be translated, interestingly enough, into biological networks. So the first thing that we discovered is, one, there were 4 major biological networks involved in this process, and that they were sequentially disease-perturbed in an invariant temporal order, starting with prion replication and accumulation, the most unique system, going to glial activation and 2 forms of neurodegeneration. The key here is you want to focus on the first perturbation, because in disease, if we can understand that and create drugs to revert the individual back to normal at the very beginning, you don't go through a process of irreversibility by the time you get to the clinical symptoms that say this animal is sick. One of the things that was really interesting is we asked, it's one thing to be able to sample the brain and get the brain transcriptome. Could we actually look in the blood and see the same temporal kind of order? And the approach that we took was to identify transcripts in the brain that are brain-specific. Then we looked at those that secreted proteins that could be detected by mass spectrometry in the blood. We identified about 200 of them. And we showed that those proteins then mapped back into the fundamental biological functions of the brain. So in a sense, these brain-specific blood proteins were assays for being able to look when a normal brain began to transition into a disease state for any of these major kinds of activities. And we used then 15 proteins that we identified in the mouse that were brain-specific that mapped into the 4 major networks and demonstrated almost exactly in the blood the same temporal order that we saw in the transcriptomes. So in the blood, we can, at the very earliest stage, detect the changes that indicate that the prion replication and accumulation network has become disease-perturbed. And of course, this opens up then the whole strategy of being able to look at whether you can modify this very first network before others became actually engaged. A second point is the one of making blood a window into health and disease. And the approach that we took initially with the company Integrated Diagnostics that we started 6 years ago was to use mass spectrometry to interrogate relevant proteins in the blood to see if we could gain a panel of proteins that had the ability to distinguish benign from neoplastic nodules in the brain. There are 3 million such nodules that occur. 600,000 of these people go to surgery. More than half the surgeries are unnecessary. So we were curious as to whether we could distinguish benign from neoplastic nodules and save the surgery. And the bottom answer is we used a series of systems approaches, starting with about 400 proteins, and then a series of systems filters to go to eliminate all of the noise that existed in those 400 proteins. And that brought us down to a panel of 13 proteins that had the ability, with about 40% success, of identifying benign nodules as opposed to their neoplastic counterparts. We, in the last year, have done exactly the same for positive diagnoses. Now, we have a diagnosis that's 90% successful in identifying the neoplastic as opposed to the benign nodules. So you can put these things together. And what this leads to is the ability to save the healthcare system $3.5 billion in unnecessary surgeries, and at the same time, it clearly improves healthcare. And this test has been widely adopted by the payers because they see it can save money. A relevant question we can ask is, what exactly do these 13 proteins do? 12 of the 13 map into 3 major disease-perturbed networks in small cell lung cancer. So they give us the ability, 1, to identify the earliest transitions that occur in this disease, and 2, to follow the progression of the disease and its response to therapy all the way through. We've used a very similar approach to look at the disease PTSD, looking at veterans from Afghanistan with and without the disease. And here we made really a striking observation. If you put together a combination of 2 different types of biomarkers, you could enormously enhance the sensitivity and specificity because you avoided enormously the overfitting that plagues these simple kinds of tests. So we got, on an average, 95% sensitivity and specificity. But we got it using 1, several proteins, and 2, a number of microRNAs. And actually, the 6th of the instruments we developed was the nanostring encounter. It's an instrument that does single molecule analysis. And it can, from the same 5 microliters of blood, analyze microRNAs, messenger RNAs, and protein ELISA assay. So a single instrument then can let you do this multiplex diagnostics, which I would predict is really going to be important in the future of diagnostics. So let's talk now a little bit about peptide-based protein capture agents. This is work that was pioneered by Jim Heath at Caltech, and a couple of years ago we started a company called Indy Molecular to carry out these analyses. And the idea was the following. Jim created a library of about 10 to the 6th circular 5-mer D-amino acid peptides, and he showed that you could take a protein against which you needed to make protein capture agents, and you could identify low-affinity monomeric circular peptides. Using a click chemistry, you could actually join together in the appropriate three-dimensional configuration 2 of those monomers to give you a dimer, or even 3 to give you a trimer. Dimers usually gave you mid-nanomolar sensitivity, sensitivity of good antibodies, and trimers were half a log better. So what these agents are all about then, interestingly enough, is one, they're incredibly stable. So you could put them in an envelope and send them to Africa, and you could demonstrate that their affinity did not change at all. Number 2, they're sensitive. You can get an increase in sensitivity with each monomer that you add. Number 3, they're digital in the sense if you identify, say, the 2 monomers of a dimer plus the structure of the linking reagent, you can put all 3 of those together in vitro with copper catalysis and assemble them. 4, they can very precisely be directed at peptide epitopes. And in doing this, you cut down enormously the cross-reactivities that at least plague antibodies when you analyze antibodies in the form of ELISA assays in very complex mixtures and so forth. And it's clear this can be scaled up to generate lots and lots of protein peptide capture agents, and I'll show you what we plan to do in the not too distant future in that regard a little bit later on. So you can do in vitro diagnosis, you can do in vivo diagnosis, and you can show that these could be really powerful therapeutic reagents. Jim recently demonstrated a beautiful reagent against KRAS, which is a molecule very hard to make reagents against. So my prediction is these will replace monoclonal antibodies in the next 10 to 15 years. And again, pioneered by Jim Heath and Indy Molecular. Now, over the last 5 years, as we heard earlier again this morning, there's been a convergence, I think, of 4 major thrusts. So one is systems medicine, which I talked about. A second is the digital revolution of self-measurements, the Apple Watch and the like. The third is big data and its analytics. And of course, the fourth are consumer-based networks. And I'll tell you why I think all of those lead to a much more detailed definition of P4 medicine. Again, predictive, preventive, personalized, and participatory. The first 3 Ps, I think, are quite clear. The 4th P is all about making the patient the center of the decision-making process for their own health. And we'll show you how we've done that in interesting ways. So P4 medicine differs from contemporary medicine in that it's proactive rather than reactive. It's focused on individuals and not generally on populations. It places a major emphasis on wellness as well as disease, not just on disease. It's about generating these dense and dynamic personalized data clouds. And it's skeptical of using the contemporary approach to clinical trials, 30,000 patients, give them a drug, give them a placebo, extract the average results from that population. And we'd argue that's wrong because each individual is genetically and environmentally unique. And what you do when you average is you increase the noise and you decrease the signal. So we'll do the same studies by looking at the individual data clouds aggregated to 5 or 10 or 20,000, and we'll segregate them according to the properties that you're interested in: response to a drug, failure to response, whatever you wish. But the important point is the essence of the analysis will come from what the individual is and not the average population. And of course, the final point one can make is I think these patient-activated social networks are going to be key for learning about the new medicine, for crowdsourcing, figure out how to optimize your health, even independently from the physician. And of course, I think they're going to be really key for advocacy because, again, the current contemporary system is quite conservative, unwilling to change. So P4 medicine then is about wellness on the one hand, it's about disease on the other hand. 98% of society's resources go into disease, almost nothing into wellness. And what is going to be key is to create metrics for wellness that let us quantitatively assess for each individual that. And that's a point that I'll return to later. I would argue that the scientific wellness industry is going to become a segment that's quite independent from the disease industry. And I think we'll see a shift of society's dollars from disease to wellness in the next 10 to 15 years. And I think at that time, the market cap of the wellness industry will far exceed that of the disease industry. And of course, what's really key to realize is that wellness gives us two powerful new abilities. One, the ability to optimize human wellness, and we'll talk about that in just a moment. And two, following wellness to disease transitions lets us identify the very earliest transitions and get the diagnostics and therapeutics that are going to be necessary for early reversal and for saving the healthcare system enormous numbers of dollars. Why is wellness really important? If you look at the statistics of the last 10 years or so for longevity, you can make the argument that half the children born in this calendar year will live to be 100. And the question is, what's the quality of their life for the last 30 years or so? And we think scientific wellness can absolutely transform that quality. What we did in 2014 was to create a pilot study to carry out the initial stage of this 100,000 wellness project that we talked about earlier. We elicited 107 volunteers, mostly in the Seattle area, to undergo dense and dynamic data cloud analyses over the period of the year, and generated from that whole genome sequences. Every 3 months, we got blood, urine, and saliva to get clinical chemistries, 400 metabolites, 700 metabolites, and 400 proteins. And every 3 months, we did the gut microbiome, and we used Fitbit to assess activity and quality of sleep and so forth. And this gave us then these dense, dynamic, individual data clouds, which, if appropriately integrated and compared against a database from the literature, we could integrate and model these things to identify actionable possibilities. And as I said earlier, these actionable possibilities, hello, anyone awake up there? Can we advance the actionable possibilities actually permit individuals to optimize wellness and to avoid disease. And what we've learned that is absolutely fascinating is that as we begin to gather more data and do the data integrations, more actionable possibilities are revealed. So this is going to be an exponentially expanding series of opportunities that emerge from these data integrations. The really critical point is we use coaches to bring these actionable possibilities to the individuals, explaining both what an actionable possibility was and putting it in the context of their own healthcare objectives. And they were 70% successful in persuading people to change their behaviors, which is really quite remarkable. And I would argue that what we're going to be able to do here is 107 N-of-1 experiments for nutrition, inflammation, and a whole variety of other things. And these N-of-1 experiments have already been revelatory in absolutely fascinating ways. So let's talk just a little bit about the data that has emerged from the 100 pioneers over a year. My colleague Nathan Price at ISB is the computational wizard behind these analyses. So I would liken what we've done in creating these dense and dynamic data clouds is akin to the Hubble telescope, which for the first time allowed us to look at the dark matter of the universe and gain insights and correlations of a very deep and specific nature. And I think exactly the same is true with all the data that we've generated from each of these individuals. Because when we've looked at the 5 major classes of data and the individual data types within it, and this is a scaled-down version of what we really have, we've been able to make 35,000 statistically significant correlations between a data type a data piece in one type of data and a data piece in another type of data. And these correlations give us insights into systems that interact that we had no idea about. They give us new actionable kinds of possibilities. They raise fascinating clinical opportunities. And they're really the substrate for biology that I think really is going to be enormously transformational. And to give you a specific example, one of the microbes in the gut correlated in a negative way with bile acid. And we did a classic text searching analysis of literature and showed that microbe destroyed bile acid beautifully. There is a disease in the 3rd trimester of pregnancy, cholestasis, where the female hormones block the bile duct, bile acid pours into the blood, your appetite goes away, everything tastes acidic, you swell up. It's a miserable 3rd trimester. Wouldn't it be interesting if we could make a probiotic of this microbe and feed it to these women dealing with the excess bile acid? So that's just a specific example of what we have. There are 10 or 11 more text searching analyses we've made out of the 35,000 correlations. We can argue that with 107 people, the statistical power is limited, although we've done 3 different statistical analyses, and they all correlate for the 35,000. But as I'll tell you later, we will, in the next 18 months, have 10,000 new dense and dynamic data clouds, and they'll really give us the power for examining these kind of things. So these correlations permit 2 really interesting things. They permit us to begin asking how these correlations lead to mechanisms that are fundamental to wellness and disease, and we'll be using these systems approaches to do that. And equally important, if you analyze the individuals at the level of those correlations, they give us an incredibly powerful ability to stratify individuals in ways they could never be stratified before with regard to responses, with regard to disease, with regard to many different types of opportunities and so forth. Second thing that we've been able to demonstrate is we can determine your genetic risk for perhaps 60 diseases, those that are associated with GWAS types of data. And I think many of you are familiar with the fact GWAS studies operate generally at the level of tens or hundreds of thousands of individuals. And Here's a study looking at almost 200,000 where 72 loci associated with cholesterol levels were identified. And what we can do, because each of these GWAS markers has associated with it a probability, is we can sum those probabilities for each individual and get an assessment of genetic risk. The GWAS markers in some cases facilitate the trait, in other cases they reverse the trait, so hence the green and red there. And of course, what is really interesting is if we bin the 107 individuals with regard to LDL cholesterol levels and so forth, from least likely to very high probability into 5 bins, you can see in the green there's a beautiful correlation between the disease phenotype, cholesterol, and the genetic risk. And you can see there the red turns down at the end. Those are the individuals that have statins. And of course, therapy has knocked down their cholesterol levels. But I'll just make a really important point. Your genetic risk has biomarkers that are associated with it that are absolutely independent of whether you have the disease or not. When you're studying biomarkers for cholesterol, you often can conflate the biomarkers for genetic risk from those with the disease itself. And for the first time, we can disambiguate those correlations, and it's going to enormously improve diagnostics and allow us to distinguish genetic risk from actual disease states and so forth. We have 2,000 normal individuals whose genomes have been sequenced at ISB, so we can carry out the GWAS study analyses on each of those, and we can create then a normal distribution of likelihood of low to high disease risk, and then we can place each of the 107 individuals on that population basis, as we've done here with 2 different individuals. So that means we can actually calculate genetic risk for the 60 or 70 GWAS diseases, and I'll show you about one that we're going to do with regard to Alzheimer's. So here's the population distributions. In most cases, they look more or less like normal distributions, but for late onset Alzheimer's, for example, you've really got a curious shoulder there that we don't understand at all. So we're actually now in a position to use the genetic markers, both the ApoE4 alleles and the GWAS markers, to identify genetic risks for individuals, and then to map those risks into a normal population of 2,000 to determine the high-risk populations. And we're going to follow 200 individuals that have high risk for Alzheimer's, looking at the earliest transitions and using systems approaches to understand how we can generate early blood diagnostics and eventually therapeutic reagents. Dale, president at UCLA, has made the argument, and somewhat convincingly, that if we can give him individuals that have the earliest Alzheimer's transitions, he has a 36 interventional perturbation approach that will delay the onset of this disease from 5 to 15 years. So we are collaborating with Dale on this kind of an analysis. And the final data I'll show you is the gut microbiome. And here are the ratios of microbes found in the gut microbiomes of the 107 individuals. And you can see more or less a continuous distribution all the way across these individuals. What we've also done is we've binned the 107 individuals with regard to Crohn's disease from very low to very high risk in 5 buckets. And then we've mapped on top of that the average gut microbiomes for each. And what I think was really striking here is there is one pro-inflammatory bacterium that has a beautiful linear correlation that increases as your genetic risk actually increases. And again, one wonders what you could do with probiotics to eliminate this microbe. But again, I'll make the point, only one of our individuals actually had been diagnosed with Crohn's disease, so you really have to separate genetic risk from markers that are associated with the disease itself once again. State transitions, we've actually identified individuals that go from less to greater wellness and the inverse, from wellness to disease and the inverse. And of course, with the actionable possibilities, we see striking changes in the dense and dynamic data clouds that are among the most positively reinforcing effects for the individuals that are involved. In the first clinical chemistries, we saw 91% of the individuals had nutritional abnormalities, almost 70 inflammatory abnormalities, and of course, cardiovascular and diabetic abnormalities as well. What was striking is 43 individuals were pre-diabetic. By the end of the study, 7 had moved to be normal, and almost all of the rest had moved significantly toward being normal. And of course, the interesting question is, why was there a different rate in conversion? And we are doing N of 1 studies to begin looking at that at this point in time. Most of the nutritional difficulties have been corrected, and much of the inflammation has been corrected too. So overall, about 100% of the individuals had perception of increasing their wellness over the course of the study. Genetic variants can determine what your most effective diet is. They can also determine what your most effective exercise is, and there are more than 300 SNPs that lead toward a propensity to various athletic injuries. So there are many ways we can give advice and counsel to the patients, and those first 2 possibilities turned out to be extremely useful for many of the individuals that we studied. One of the individuals that we studied came in with arthritis that had plagued him for 6 to 12 months. He'd had a concierge physician on the East and West Coast. They hadn't figured out what was going on. When we got back the clinical chemistries, his transferrin levels were very high, meaning he had a high level of iron. When we looked at the genetics, he was homozygous for the disease hemochromatosis. And of course, what was interesting is that's an easy, actionable possibility in that you draw a unit of blood every month until you bring the iron level down to normal. And by the end of the study, his arthritis had reversed completely. So it was a spectacular success of early disease that got reversed back to normality. So as you can see, we've got fascinating statistical correlations that let us think about mechanisms, that let us think about patient stratifications. We've got this correlation between genetic risk and disease phenotype for 5 different examples, 5 out of 5 examples we've looked at, which make us believe we can do genetic risk predictions from GWAS data. And I haven't talked about this, but we can analyze in individuals that go from less to greater wellness, both protein and metabolite changes that are beginning to give us a metric that will allow us to assess wellness in each individual. The insights that the pioneers had, I think, were really interesting. It was, for many, an experience of a lifetime. They most all felt healthier after they were finished. They had the idea that it was wonderful. They could take control of their own health and make appropriate decisions with the proper data. They realized that genomes don't control your destiny, just your potential, and you can circumvent many genetic limitations with behavior changes. We're all less well than we think we are. I would say if we set a scale from 1 to 100 of wellness, I'd say a lot of people in this study were 50 or 60 and below, and relatively few were up at the higher end. 70% of the actionable possibilities were indeed acted upon. virtually all the pioneers wanted to continue in the next phase of the study, and that takes two directions. We started about 3 months ago a scientific wellness company called Aerovale, focused at consumers, and we've brought many of the 100 pioneers into this study, and it is going to be programmed on exactly what we did in the 100 pioneer study with modifications. But the idea that's really exciting is we plan to get 10,000 dense dynamic data clouds in the next 18 months, and that ISB is going to play a major role together with Aerovale, the company, in analyzing all of those data in ways similar to what we've already discussed. Another area is ISB itself will seek partners for smaller studies where we'll apply this longitudinal dense dynamic data cloud approach both to clinical populations where we want to optimize wellness and to disease or populations at risk for disease where we can follow wellness to disease transitions or the response to therapy and so forth. So I think these are going to be very powerful approaches in the future. And I won't go through and list all the places where I think scientific wellness and precision pediatrics could intersect, but I think there are many. I mean, one that's always intrigued me, suppose we took mothers who wanted to get pregnant, we optimized their wellness, we followed them and the fetus all the way through, and then the fetus afterwards, comparing it with populations that weren't so optimized. And I think looking at epigenetic differences would be absolutely fascinating in this regard. And following mother, father, infant trios through pregnancy to look at various diseases of pregnancy, looking at normal infants to follow their wellness, sick kids to follow treatment and their response and so forth, and doing longitudinal studies on all kids, I think would be absolutely fascinating. And of course, you can follow infants with difficulties, failure to thrive, genetic defects, and so forth. One of the real constraints in applying this to children is we do use a fair amount of blood in the adult studies. So a proposal that we're thinking about now is using microfluidics and the organ-specific blood proteins from 50 different organs to make measurements either in adults or, in this case, children. So with Jim Heath again, we've actually got a device where we've done 50 ELISA assays commercially available now. It takes a fraction of a droplet of blood. It does it in 5 minutes. High sensitivity, and it's already been used very nicely in hospitals. And we think we can easily scale this from 50 to the 2,500 blood proteins you'd want to measure if you got 50 organ-specific proteins from each of your 50 major organs. And of course, that would let you follow wellness to disease transition straight away for very small amounts of blood. You could easily do it once a month or so. So transforming healthcare, we've defined many new actionable possibilities. We've shown that the actionable possibilities can improve health and or let an individual avoid disease. We're beginning to get metrics for wellness now that will let us make objective quantitative measurements in the future. And we're beginning to study wellness to disease transitions to look at the very earliest stages of disease and think about how to have an early transition back to wellness. We're going to drive the development of assays. We have to miniaturize the assays for blood and other kinds of samples, but we'd really like to drive down the cost of the assays, which is a limiting factor at this point in time. And that really is what the digitalization of medicine is going to be all about, the creation of assays that will follow a Moore's Law decline in costs. And I would guess in a 5-year period, the cost will be 10% or less of what they are today. We're going to have a database that measures well individuals and disease transitions, and it obviously is going to catalyze innovation for the wellness industry as a whole. And I'd argue that this is P4 medicine that we're beginning to bring to the healthcare system, improving the quality of healthcare, decreasing costs, and really promoting innovation. So my vision for the future of healthcare is, one, I think the P4 paradigm is going to be a dominant paradigm in the future of healthcare. I think number two, health really is all about assessing the environment, your environment and your genetics, and of course the dense dynamic data clouds let us do that beautifully. And this is how we're going to be able to optimize health, wellness in the future. Wellness is going to do the two things I talked about earlier: optimize human potential and capital by maximizing wellness, but it's going to give us the transitions, wellness to disease, so we can again revert those back to normal at an early stage. I think 10,000 dense dynamic data clouds is utterly going to transform how pharma, biotech, nutrition, and diagnostic companies think about carrying out their acts. And we can give you 3 or 4 or 5 reasons why that's so. And in the same vein, I think Aravel is really going to be pushing scientific wellness, driving down the costs to the point sometime in the future where we'll be able to take scientific wellness to the poor as well as to the rich. And this will lead to a democratization of healthcare, inconceivable even 5 years ago or so. And I would say the last point I'd like to make is scientific wellness, in interesting ways, I think, can deal both with aging and with the end-of-life expensive costs. And the end of life is, if you die over a period of 2 or 3 years, you can spend 50% of your health care dollars during that time. So there are 2 interesting observations. One is Eric Topol at Scripps Institute has 1,000 individuals 90 or older that he calls the welderly, that have never been sick, never taken a drug, are in superb health. Observation 2, we did a study some years ago about 18 individuals, 115 or older, to complete genome sequence analysis, and we just didn't have the statistical power to make any very firm conclusions. What we did learn is if you get to 100, almost always you die very rapidly due to a complete systems failure. So I think scientific wellness is really going to be a lifelong journey, and if individuals are willing to act upon their actionable possibilities, I think we have a real possibility of converting them operationally to the welderly, push you into your 90s, alert, physically active, and the like. And of course, if we can get you to 100, then we'll leave you on your own to deal with the complete systems failure and everything. Anyway, I just want to make the point, I think we're looking at an enormous revolution. I think we're looking at an enormous opportunity for institutions that are willing to dive in and embrace the revolution that's coming. Thank you very much.